What I do is I mostly focus on chemoproteomics and using chemoproteomics for target identification and finding the reactive sites within the across the proteome. In the chemoproteomics, we are trying, these are covalent small molecules, and we are using chemoproteomics for target identification. A lot of time these are target agnostics, and also to find to profile the selectivity of whole proteome selectivity of these compounds from the covalent aspects of it, but also also proteomics is used to profile the degradation profile of these compounds. I've been using the trypsin from Promega from long time ago. When I started the proteomics, I was using trypsin from different vendors, including Promega, and I found out how it's a really high quality trypsin. And then later on, I was using the trypsin lyc and was benefited from the lower miscleavage from mixing these two, and that is one of the proteases that really helps our work, that boosts the quality of our data. But specifically for chemoproteomics, the difference between the general proteomics field is that here we are focusing on reactive sites versus proteomics that are looking at the whole proteome. When you're looking at the whole proteome, you can always find trypsin cleaved peptides within the proteome, some peptides that are well cleaved by the trypsin, and they have a good ionization efficiency for mass spectrometry, right? So there would be a few peptides from each protein that you use for quantification. But when you are in chemoproteomics, you are interested in a certain peptide in very specific sites within the proteome. And if the sequence of the amino acid sequence in that specific site is not well behaved as a trypsin, if you, for example, don't have a trypsin site, for example, a lot of these kinases, their catalytic domain is so hydrophobic, you can't find a lot of arginine and lysines there, right? So the peptides become big and so hard to detect them with mass spectrometry. So these inventory of different proteases that I can find in Promega is really helping us to find the right protease for that specific site that we are looking for. And that is something that is I think probably every proteomic workflow can benefit from it, but specifically in our field, that is even more important, and it's really helping. To characterize the protein degradation was really interesting to me, and the diversity of it, because I wasn't as much familiar with it. One of the very interesting ones was from one of your colleagues from Promega, using the nanobreath assay for characterizing the permeability and availability of the compounds inside the cells, because I'm using PAMPA a lot, right? So, and I'm familiar with the limitations that PAMPA has for finding the permeability, and using this assay was really interesting, and it's kind of show me a new perspective of how to do permeability inside the cells.